Hello everyone, my name is Isabella Hazelton and I am a biology student here at the University of North Carolina at Chapel Hill. Today I will be explaining a molecular technique known as immunohistochemistry, or IHC for short. So I'm sure you're probably wondering what exactly is immunohistochemistry and what's it used for. Well, if we break down the word into three smaller words, we can determine what it is used for. Immuno relates to the immune system, which involves proteins called antigens and antibodies. Histo means tissue, which is what IHC examines. And lastly, chemistry involves the binding of molecules, which we will see in this technique. Thus, immunohistochemistry equals antibody tissue binding. Remember this for later. Now, IHC is used in both clinical and research settings. In clinics, IHC can be used to diagnose conditions like cancer. In research, IHC is used to determine where within a tissue a certain protein or antigen is expressed. A lot of labs here on campus use immunohistochemistry for their experiments. If you want to know what immunohistochemistry looks like, look at the right-hand portion of the slide. This is the final product of immunohistochemistry. So now that we know what immunohistochemistry is used for, let's examine how it's performed and how we begin. First, you will need to collect your tissue sample and slice it into smaller sections using a microtome, which is an instrument that scientists use to cut very thin slices of material. So if you look on the right-hand portion of the slide, you'll see a slice of brain tissue, and right next to it on the left is a microtome, which is what we are going to use to cut that brain tissue into smaller pieces, which allows us to perform IHC. After this, you perform a step known as blocking. In immunology, antibodies bind to antigens that are located on the surface of a cell. So in the blocking step, a blocking agent is used to prevent antibodies from accidentally binding to an antigen or protein that you are not interested in. Now that we've determined how to begin preparing for IHC, let's see how we actually perform this kind of experiment. So we're going to be getting into the nitty-gritty of IHC. First, you are going to add your primary antibody, which is designed to specifically bind to your protein of interest. We don't want it to bind to any other protein besides your protein of interest. After this, the washing step utilizes something called a buffer solution, which will wash away the excess antibody that hasn't bound to the protein of interest. Next, you will add your secondary antibody, which has been labeled with a marker molecule. This marker molecule might be a fluorescent dye or an enzyme. Unsurprisingly, the secondary antibody labeled with a marker molecule will bind to the primary antibody, much like how the primary antibody binds to the antigen or protein, allowing scientists to better visualize and amplify the signal of the protein. Lastly, the washing step is used again to wash away excess secondary antibodies. So basically, to summarize, if you look on the right-hand side of this slide, we have our piece of tissue, we have our protein of interest, which is bound to the surface of the tissue. We have our primary antibody, which binds to the protein of interest specifically, no other proteins. It doesn't bind to any other protein. Then we have our secondary antibody, which will bind specifically to our primary antibody and no other antibodies. And then we have this little fluorescent marker, which is labeled onto the secondary antibody. And that allows us to just visualize where our protein is expressed. So now that we have performed our experiment, how do we analyze our results? First things first, if you used an enzyme as your molecular marker, you're going to need to perform a chemical reaction. This chemical reaction involves adding a substrate, which is just a molecule that will bind specifically to the enzyme in the mixture, which you used as your fluorescent marker, resulting in a chemical reaction that produces a visible color change. After you have done this, you can use a microscope to examine your tissue. And that's it for IHC. Thank you so much for watching. I hope you enjoyed your science lesson.